Bone is very different from cartilage, and it differs by being mineralized, is highly vascularized, and has extensive nerve supply. In addition, it's also very dynamic, which means that it's able to remodel and repair, unlike cartilage. It also interacts with other tissues such as the muscle and ligaments. Function of bone is to support our cells. It also protects the vital organs within, and it also acts to provide locomotion by acting through tendon and ligaments. It's also a reservoir for mineral, and for that reason it's involved in homeostasis of mineral within the body. Bone cells are also supportive of hematopoiesis. So bone marrow is consistent with hematopoiesis through the stroma that's provided by bone cells. Composition of bone is mostly inorganic matrix, which is hydroxyapatite, about 70% by weight. 8% of bone is organic matrix, 22% by weight is water. So bones are not really dry, as some may think. Now, if you were to look at the organic matrix itself in this pie chart, you can see that most of that organic matrix is collagen or collagenous, and is exclusively type 1. We have some non-collagenous component, about 5%, and then 5% is cells. The organic matrix, as we said, is mostly type 1 collagen. We also has GAGs and proteoglycans, and some non-collagenous proteins, including osteocalcin, bone solid proteins. Again, this list is not exhaustive. There are three types of bone cells that you should be able to identify. So there are osteoblasts, which are forming bone, osteocytes, which are mature osteoblasts, as well as osteoclasts, which are involved in resorption of bone. So osteoblasts are derived from primitive osteoprogenitor cells, which themselves come from mesenchymal stem cells. Osteoblasts are located on the inner bone surfaces as well as on the outer part, which you will see underneath the periosteum. The word periosteum would be defined a little bit later. Osteoblasts are involved in secreting the organic matrix of bone, which is called osteoid, which then becomes mineralized, which gives rise to the classical extracellular matrix of bone. Once they have finished their work, they become osteocytes by being surrounded by the mineralized matrix, which we will see a little bit later. This histological view of developing bone demonstrates osteoblasts very nicely. These are these cells lining the surfaces of the bone, and you can see them. They are plump because they are actively producing the osteoid, which is seen as this lighter color staining surface right underneath them, and which then eventually becomes mineralized and become darker, as you see it here in the middle portions here. One thing to note here is that this surface here is lined with actively synthesizing osteoblasts, whereas on the other side, as you can see, those cells appear to be missing, but they really are quiescent. They are just sitting there, not producing the matrix, but they are there, and they are usually referred to as lining cells. Now the osteocytes, as we said before, are the mature osteoblasts, and they are surrounded by the matrix they have themselves produced. One of the things that they have very critical in terms of their function is this extensive cytoplasmic processes in canaliculi. So let's define what we mean by cytoplasmic processes. Cytoplasmic processes are like the extensions of the cells themselves, which are extending out from the cells. And then canaliculi are the channels within the matrix through which these processes are passing through. So if the adjacent cells have these processes, they effectively talk to each other through these processes by joining together with gap junctions. So that's how the osteocytes are able to communicate with each other through the mineralized matrix, through the gap junctions. These cells are very important in bone turnover, maintenance, as well as bone repair by being mechanosensors, and the process through which they act is referred to as a mechanotransduction. So these cells are able to sense forces on bone and then communicate out to the cells which are able to either form or resorb bone. So this is a very nice picture, histological view of how the osteocytes are formed. So in this case, you can see this darker area here is the mineralized matrix. The osteoblasts are sitting on the surface here, producing the osteoid, in this case quite a thick seam of the osteoid. So if you were to look at this cell here, as you can imagine, it was sitting on the surface a little while ago, and now it's becoming surrounded by the matrix it has synthesized itself. And then this cell is temporarily a little bit earlier, so now it's completely surrounded by the matrix. Note that this matrix is still unmineralized, so this is still within the osteoid. So this cell here is a little bit earlier in time, so now it's getting surrounded by the mineralized matrix. So this osteoid is going to be inundated with this mineral very soon, and this cell will become bona fide osteocyte. So these EM pictures are a clear demonstration of the cell processes that we spoke about earlier. So here's a cell which is sitting in its little space here, which is referred to as lacunae, and you can see these processes coming out. And if you look at these two cells, the processes from each one of them are making junctions right around here, where this cell can talk to this cell. But these processes also allow the individual cells to talk to cells in three-dimensionally, to all the cells surrounding it. This picture here is even better demonstration of the network of processes. Here's a cell, here's an impression of another cell down here, and then this cell, this osteocyte, is very close to the surface of bone. As you can see here, that's the surface of bone, and these processes are able to then pass the message over onto the outside, where the osteoblastic or osteoclastic cells will be sitting, and they can hear what the cells within the matrix are saying through this particular cell here. So third type of bone cell is the osteoclast, and this cell is specialized in resorbing bone. It's a fairly large cell. It can be close to 100 micrometers in diameter. Characteristically, it has multiple nuclei. So let's be clear about that. So this cell is actually, it contains individual nuclei which are in number are more than one and could be as many as 200. They are derived from these pluripotent cells of the hematopoietic origin. So it's referred to as the cells coming in from the monocytic cell lines, and the osteoclasts are formed by the fusion of monocytes, and thus explains these multiple nuclei within one cell. They are not the same as regular macrophages, although their function is similar, but they are more specialized in that they not only phagocytose the material, but also degrade the material externally to themselves. So we can refer to them as bone macrophages, but they are not typical macrophages. So histologically, you will see cells which are on the surface here and making indentations into the bone, and you see these multiple nuclei. In this plane of section, you don't see that many. But if you look at this picture here, you can see an osteoclast eating or resorbing bone here, has multiple nuclei here, and even this cell here, for example. So these cells are able to resorb large amount of matrix because of their size.